Good afternoon and welcome to our research briefings. Every year we take this opportunity to hear talks by newly elected members from each of the 6 NAS classes. Think of this as a sampling. The speakers represent their sections and classes, but we've asked them to focus on their own careers, telling us about their science and how they came to be scientists. All were formally inducted yesterday evening. I think you're going to find this session both enjoyable and enlightening. The first speaker is Flaminia Catteruccia, the Irene Heinz Given Professor of Immunology and Infectious Diseases and a Howard Hughes Medical Institute investigator at the Harvard T.H. Chan School of Public Health. She received her undergraduate degree from Sapienza in Rome and her PhD from Imperial College London. Dr. Catteruccia studies malaria, a parasitic disease transmitted by mosquitoes that has afflicted humanity throughout our evolutionary history. It's still prevalent across large swaths of our planet, killing hundreds of thousands of individuals each year, including many children in sub-Saharan Africa. Dr. Catteruccia's group studies factors that influence interactions between the mosquitoes and the parasites and consequently influence disease transmission. Her work to find novel approaches to reduce the transmission of this devastating disease is becoming even more important as climate change increases its prevalence. Elected to Class 6's section on Animal Nutritional and Applied Microbial Sciences, Dr. Catteruccia's title is What's the Buzz About Malaria? Novel Solutions for Blocking Transmission of an Ancient Pathogen.
Thanks so much, Nancy, and I'm so honored to be here today and represent my class. And I want to thank the Academy for giving me the opportunity to talk about the path that has taken me to where I am today. And I'm going to start to describe my path by talking about my first name, Flaminia. Perhaps not many of you here know that Flaminia is an ancient Roman name, and so this name says a lot about me and my upbringing, because actually I was born in Rome, and my parents gave me this name because they were also born in Rome and because they loved this city, and they really instilled in me and my sisters a passion for this city. And because of this, because I was surrounded by such incredible beauty and history, growing up, I really wanted to be an archaeologist. And this was somehow supported my father, who gave me this book about the discovery of Troy by the archaeologist Heinrich Schliemann. And Troy was the site of this legendary war between the Greeks and the Trojans that was so vividly depicted in the Iliad and the Odyssey that I was studying at school at the same time. However, I also liked science a tiny bit. And so when the time came to decide what to do in college, I decided to take a completely different path, and I decided to major in organic chemistry at La Sapienza. And this is because I was also a pragmatic kid, and I knew that a degree in chemistry was likely to give me a job at the end of my studies. And that's exactly what happened. As soon as I finished, I was offered jobs in pharmaceutical companies around Rome. But of course, I didn't want to work in industry anymore. I was young and I wanted to explore the world and be free. And so I decided to do something completely different, and I moved to London, where actually I did my PhD at Imperial College in the lab of Professor Crisanti to study the malaria mosquitoes, the Anopheles mosquito that transmit malaria. And after all, malaria is a very ancient disease that has afflicted humanity since our very first beginning. And moreover, malaria and Rome have a long history that spans millennia. In fact, famous people like Julius Caesar, the general, are believed to have contracted malaria at some point during their lives. And moreover, let me also indulge you in a bit of gossip. Caesar's political ally on the other side of the Mediterranean, and presumed to be lover Cleopatra, Queen of Egypt, is believed to have protected herself from mosquito bites by sleeping under a mosquito net that was actually made of golden threads. So, very glamorous. And I don't know whether this is real or it's fake news. I mean, fake news are so popular at the moment anyway. But what's true for sure is that neither Caesar nor Cleopatra could have known that the disease that was affecting so many people around them was caused by a specific genus of mosquitoes called Anopheles. And it took humanity about 2,000 years for this discovery to be made, and it was made actually in Rome by this very famous scientist, Giovanni Battista Grassi, who was studying malaria patients in Roman hospitals, and they discovered for the first time that the human malaria parasites could only be transmitted by Anopheles mosquitoes. And unfortunately, he did not get the Nobel Prize for this discovery, mostly for political reasons, and the Nobel was given to his colleague, Ronald Ross, for his studies on transmission of bird malaria. However, the studies of Grassi really laid the foundation of malaria research as we know it today. And so what is malaria? Well, as I mentioned, malaria is a disease caused by Plasmodium parasites, which to this day still kills hundreds of thousands of people, especially young children in Africa. And because malaria relies on mosquitoes for its transmission, historically we've been targeting the mosquito for malaria control. And last century we believed that we had the silver bullet when DDT was discovered as a potent insecticide and that could kill mosquitoes. And so we thought that DDT would eradicate malaria from the face of the world. However, we all know how it went, and a century later, we still have malaria, we still have lots of mosquitoes, and we are still using insecticides to control this disease. Insecticides are sprayed on house walls or incorporated onto mosquito nets. Not as glamorous as Cleopatra's nets, but still meant to protect people while they are asleep. And these two strategies have worked for a while and have provided a very strong contribution in the decline in malaria cases. However, after a while, mosquitoes become resistant to the action of the insecticides. They're not killed anymore. And in fact, in recent years, we've seen that the number of malaria cases is not going down any longer, also because of insecticide resistance in Anopheles mosquitoes. And so it's pretty clear that we need innovation in malaria control. And in my lab, we believe that innovation can only come from a better understanding of the biological, ecological, and environmental components that shape malaria transmission. And so when I started my independent career at Imperial College, London, and then I moved to Harvard later, I decided to focus my studies on an area of research that I thought could provide innovation in malaria control. And that area of research was mosquito sex. And mosquito sex is a very fascinating phenomenon that happens in flight, happens in swarms, where males get together at sunset when things are romantic, and then females get into the swarm and they choose a male. And the mating couple lives while still in copula, as you can see in this photo taken by my collaborator Rob Shaw during one of our many trips in Burkina Faso, where we've been studying these mating swarms in collaboration with Abdullah Diabate and his group from Bobo Dioulasso. And so this is a very dynamic event that happens in flight, so it's very energetic, and males and females are together for only 17 seconds, which I believe is short even by human standards. But this short mating event completely changes the biology and behavior of females. In fact, females will never mate again. They have sex only once, and that's it. And so obviously, for me and for my group, this was like a potentially good target for preventing mating from happening and reduce the number of mosquitoes out there that can transmit malaria. And so we were lucky enough that we could recapitulate swarming and mating behavior in our lab in London and then in Boston. And here you see a video of a mating swarm, and where on the bottom right, you'll see a male and a female mosquito getting together and then slowly leaving the screen to have some privacy. And so here you can see that this is a smooth event that goes very nicely. However, I mentioned at the beginning that females choose a male in the swarm. And you don't have to take my word for this. Actually, I have video evidence. And evidence is here. In this circle, you see a male approaching a female. The male is at the bottom, the female at the top. And at the beginning, the female seems interested, but at some point she turns and she kicks the male away, and she goes back and joins the swarm to find a better male. And this is on a loop, so we can watch it as many times as we want. And what we find is that females pick males based on how they taste, and that males that have a larger composition of specific molecules called hydrocarbons on their cuticles are those that are preferentially chosen for mating. And so this is potentially something that could help us with new malaria control methods. However, for a very long time, what we wanted to know is why mosquitoes have sex only once. What happens during that mating event that completely switches off their willingness to mate again? And studies in Drosophila, the fruit fly, by several labs had shown that during mating, males transfer small peptides called sex peptides to the female that trigger a series of events that induce refractoriness, as we call it, to further copulation. However, over the years, we demonstrated that Anopheles mosquitoes use a different system, whereby males transfer high levels of steroid hormones. And for a long time, we believed that this steroid hormone was the one shown here in orange, 20-hydroxyecdysone, or 20E. But this was puzzling for us, and it was puzzling because also females produce this hormone, 20E, every time they feed on blood, and they need this hormone to produce eggs and reproduce. So it was puzzling for us that the same hormone that females produce every time they feed on blood would also be the same hormone that males transfer to make them refractory to copulation. It seemed very risky for the species, for the survival of the species. So for a while, we postulated that the hormone transferred by the male must be different from 20E and must be a male-specific or predominant hormone that male specifically transferred during copulation. However, we could never find this hormone, and also male predominant hormones were not known to exist in insects. But eventually we proved that our hypothesis was correct, and we identified this 3D20E shown here in blue, which is a male predominant hormone that is very similar to 20E, it's just oxidized at carbon 3, and that males transfer in very high levels to the female. And what we have found is that this system is very intriguing because males produce high levels of this hormone in their reproductive tissues. However, in males, this hormone is inactivated because otherwise it would be damaging for the fitness of the male. And it's inactivated by phosphorylation. And males transfer the inactive form to the female during mating. However, very cleverly, they also transfer the enzyme that is important for the activation of the hormone. And so slowly over time, 3D20E is released in the female reproductive tract, and this in turn induces refractoriness to mating by mechanisms that we don't yet understand, but we are actively pursuing in the lab. So overall, these studies show that also insects, or some insects at least, possess male predominant hormones, like we do. And what's also interesting that we found is that not all Anopheles species use this mechanism and actually have males that produce steroid hormones in their reproductive tissues. In fact, when we surveyed a number of Anopheles species from all over the world, what we found is that only the species shown here in red are species in which males produce high levels of steroid hormones. All the other species have negligible levels. And you will notice that these are species from Africa, where also malaria, a lot of malaria cases are happening, and these are the species that have been transmitting malaria since really the beginning of humanity. So this is one of the reasons why we thought that we should also start looking at a possible function of steroid hormones in malaria transmission. And that's something that my group started doing when we moved to the Harvard School of Public Health in the department of Professor Diane Worth, that has also been an amazing mentor for myself and for everyone else in my group. And so indeed, what we found is that steroid hormones produced by the female, so the same hormone 20E that I showed you earlier, somehow modulates the development of Plasmodium parasites in the mosquito. In fact, if we impair the function of steroid hormones, what we see a reduction in the number of eggs, so reproduction of the mosquito is reduced, but we see also a reduction in the number of parasites that survive in that mosquito. So there are fewer parasites. Parasites somehow die when we reduce the reproduction of females. So this seems like great news. So we can have fewer mosquitoes, and moreover, those mosquitoes have fewer parasites that they support. So it seems like good news for malaria transmission. However, biology is never that simple. And what we found by looking at those parasites is that although they are fewer in numbers, they look larger, they're bigger. They are actually growing faster. And what does it mean that they grow faster? It means that they get to this stage shown here, where there are thousands of these elongated parasites, which are called sporozoites, that are formed in the mosquito. And these sporozoites are the parasites that infect humans. So they are the form that mosquitoes inject into our bloodstream and cause infection. So as you can imagine, having a mosquito that produces sporozoites in a faster amount of time is not really what we want for malaria control. And so this is one of the reasons why we thought maybe targeting reproduction is not such a good idea. And so we started thinking about potential other intervention. And that's what led us to a new idea. In particular, what we wanted was an intervention in which we could prevent mosquitoes from transmitting malaria parasites without really affecting the mosquito itself. And so let me explain what our idea is. If we look at our malaria control methods at the moment, mosquitoes, we kill them. We use bed nets that incorporate insecticides. So a mosquito lands on the net, absorbs insecticides, and is killed. And this obviously induces strong selective pressure on the mosquito to develop resistance. In humans, we use drugs that kill parasites in circulation, and this obviously induces a selective pressure on the parasite to develop resistance. And in fact, we are seeing drug resistance is a major problem in malaria control. So our idea was very simple. Why don't we merge these two strategies into a single strategy, and we deliver antimalarials to the mosquito by mosquito nets. So the mosquito will land on the net, will absorb the antimalarial. The mosquito will not be killed because the antimalarial is specific for the parasite. However, parasites will be killed, and the mosquitoes will be harmless. There will be no transmission. So this seemed like a crazy idea, but instead it worked. And a few years ago, we provided proof of principle that this system works very nice. And so if we expose mosquitoes to antimalarials, those mosquitoes have zero parasites in their midgut. So there is no parasite developing compared to lots of parasites in mosquitoes that are not exposed. And moreover, very, very recently, we conducted a screen for several compounds with potent action against the parasite in the mosquito. And we found compound combinations that work very, very well, that are very active at killing parasites. And why do we want to use combinations? We want to use combination to slow down the parasites from developing resistance, because the parasite will have a very hard work at generating resistance to two separate drugs. So we want to use combinations of compounds. And so where are we at the moment? We are pushing this strategy from the field to the lab with lots and lots of collaborators, including Fitsum Tades from Ethiopia, where we are incorporating these antimalarials onto mosquito nets. And the idea is to give this net to people that live in malaria endemic areas and see whether they can make a difference in their chances of getting this disease. And so we are very excited about this part of the project. So I hope that I have convinced you that innovation in malaria control is feasible, and it's feasible by studying the biology of the transmission cycle. And I want to conclude by really thanking the people that are driving this innovation in my lab. And these are people in my lab that come every day with their passion and their energy, their enthusiasm, and their knowledge, and are working on this and so many other projects. And so it's very important that as a community, we'll do everything possible to preserve the chances of these people and so many other people in the United States and beyond to provide a crucial contribution to global health and society at large. I also want to thank the numerous collaborators that it's such a pleasure to work with, that are really helping us push these projects forward. And also, obviously, our funders without whom none of this research could happen. And finally, I also want to thank all the people that have been in my lab at some point during their lives and that have helped me with my science and also have helped me become a better mentor to them and others. And thank you for your attention.
I think we have time for a couple of questions if people want to go up to the microphones. If you do, it'd be terrific if you could identify yourself and then we'll go from there.
Wonderful talk, Dominic Ryan. When you are worried about resistance, the maintenance of a high enough exposures of the drug is really going to be important. So when you look at the combinations put onto the nets, have you looked at whether or not if they drop below a certain sustained concentration, you then generate resistance? And can you maintain the nets in a sufficiently high concentration coating?
Yeah, those are great questions. Obviously, the issue of resistance is a major problem and a major challenge for any control program in malaria. And eventually, the parasite will find a way to develop resistance to the drugs that we are using against them. But one of the reasons why we are optimistic that this will be slowed down is that apart from using two compounds with different modes of actions, which obviously will help, is also the fact that the targets that we have at the moment are targets that are essential for the parasite during mosquito stages, and any little mutation in those targets is not, parasites cannot bear it, cannot actually do with mutations to the point that they don't develop in the mosquito when they are mutated. So like, for instance, if those mutations emerge during the cycle in humans, those mutations cannot be transmitted in the mosquito. But definitely the idea is to incorporate onto bed nets enough drugs that they can survive for 3 to 5 years. That's what normal bed nets they have to have activity for at least 3 and more years. So the idea is to incorporate enough of those compounds to achieve this goal. And what's interesting is that the compounds that we're working on, we are also very aware of costs. And so we are working with compounds that are very cheap to manufacture, and so we can incorporate larger amounts and go around the problem of resistance.
And on the other side, please.
Thank you. That's a wonderful presentation. I come from Kenya, where we have malaria. And I'm really curious to know how the different mosquitoes behave, because I know in Kenya we have what we call the highland malaria and also the lake region malaria. Do those mosquitoes behave the same? Or have we been able to determine that? Yeah, because we know the lake region malaria is difficult to treat than the highland malaria. Thank you.
Yeah, thank you for the question. So what I presented you today is a strategy based on incorporating antimalarials on mosquito nets. And this can only work if mosquitoes bite at night, which is when people are asleep. However, we see that different mosquito populations, because of the pressure imposed by insecticides, also started biting earlier during the day, like when people are outdoors, and so they are not protected by bed nets. So in that case, a strategy based on the use of bed nets would not work. However, this is, the incorporations of these drugs doesn't need bed nets, can also be used for house wall sprays or on clothes or anything else. So it's not restricted to the use in bed nets. So in a way, this can be tailored to different populations of mosquitoes that have different biting and resting behaviors.
And last question on, back to the other side.
Julius Lux, Czech Academy of Sciences. So what is the role of the microbiome? I vaguely remember that there was a notion that maybe the refractoriness of certain mosquitoes to malaria is influenced by their microbiome. So what's your view about that?
Yeah, that's a very interesting question. So there are so many factors that influence the ability of mosquitoes to transmit malaria. And in fact, not every mosquito species can transmit malaria, and even among Anopheles mosquitoes, some are much better than others. And so it's a combination of factors. It's definitely genetic factors, but it's also ecological and environmental factors. And the microbiome has been shown by multiple researchers to play a role in a way allowing malaria parasites to develop. Sometimes a competitive roles against malaria parasites for resources. So that's something that is many groups are studying, and actually many groups are trying to use bacteria to kill parasites in the mosquito and prevent transmission. So that's a quite broad area of research.
Thank you very much.